Good morning, everyone. Today we are going to cover the part 3 of the human alimentary canal. The learning objective is still the same. We're still going to look at the main functions of the parts of the digestive system. Before we get into that, let's do a recap of previous lesson. So we looked at the function of the enzyme renin, and its main function is to coagulate and curdle milk protein. This enzyme can be found in the stomach, especially in young animals and young humans as well. What coagulate and curdle milk protein means that the milk protein that is soluble in the liquid milk is going to become insoluble, and this enzyme helps to speed up that reaction. And this is what casein is. So casinogen is the soluble milk protein, and after the enzyme renin acts on it, it's going to kind of clump up together to become much more bigger molecule. So this will become more insoluble in water. So this is called casein now. So the main purpose of this curdling milk, it is really good for babies because it actually remains long enough in the stomach to be digested by pepsin. So pepsin in the end will digest this, but because it's a large surface area here, so it's going to take more time. Usually we want less time so that we can quickly digest things and we can quickly absorb things. But here, because babies, they don't have access to food all the time, so this will be really good in keeping like a constant supply of protein in their body. And also, by the way, this curdling of milk is actually somewhat similar. Actually, it is a process of how we try to create cheese as well. So this enzyme renin, they used to take this out of animals to actually make cheese, but we don't do that anymore. We have other things that we can use. Okay, so we also looked at the function of duodenum and the pancreas. And this is like quite elaborate. So the duodenum is I call it the C-shaped part, but it's actually called U-shaped part. I guess it's like a U too. And this front part is the duodenum. It's actually the first part of the small intestine, and it comes right after the stomach. It's also has like muscular walls, so duodenum actually has two main processes. And then we also looked at the pancreas, which has this kind of like leaf shape, but in real life it looks kind of a bit much more like rounded than like that. I think I showed you a picture previously. So the duodenum, its main function is peristalsis. So it also is made up of muscular walls, so it helps to move the food down the digestive tract, but its main function is actually digestion. So the digestion part of the duodenum is the fact that the duodenum actually receives 3 types of digestive juices. So let's go down to this line here. So the first digestive juice is called bile, and the duodenum gets this via the bile duct. This bile contains these things called bile salts and bile pigments, and bile salts is in charge of emulsifying fat. Emulsifying means to basically physically break down the large fat globules into smaller fat droplets. So this is actually physical digestion, or aka mechanical digestion, and bile salts actually does this. And the good thing about having smaller fat droplets is that it increases the surface area for enzyme, especially lipase, to act on it, so we can get a lot of fatty acids and glycerol into our body to be absorbed. Bile pigments are alkaline in nature, so they help to neutralize the acid found in the chyme. And you can see here that the pepsin that works in the stomach has an optimum pH of around 2, whereas trypsin that can be found in pancreatic juice works around 8, I think, pH 8. So this bile pigment helps to neutralize the acid found in the chyme because chyme actually comes from the mixture of the bolus after it turns in the gastric juice. So the optimum pH environment for other enzymes will be provided by the bile pigments. There's a lot of enzymes that you can actually find in the duodenum. The first set comes from the pancreatic juice. So pancreatic juice contains pancreatic amylase, which actually acts on starch, and it breaks it down into maltose. Starch is a polysaccharide, so it's really big. You can see that there's a lot of glucose molecules there chemically bonded together. And maltose is a disaccharide, so it's made out of 2 sugar molecules and it's made out of 2 glucose molecules. Trypsin is actually a protease, but this one comes from the pancreatic juice. That's why the pH is actually different than the protease found in the stomach, which is pepsin. Trypsin also acts similarly, which is that they break down protein to form short polypeptide chains. Lipase is also found in pancreatic juice, and this acts on fat. So fat molecule actually is made up of fatty acids. So these 3 is the fatty acid, and that one big rectangle right there, that's glycerol. So what happens after lipase acts on fat is that you'll break this apart and you'll get fatty acids, which is those 3, and glycerol being separated. Intestinal juice comes from the walls of the duodenum itself, and there is 1, 2, 3, 4, 5 enzymes, but we didn't actually look into this, so we'll look into that later. But just to explain, we all know that lipase, how it works, so this is pretty much similar, but we call that intestinal lipase instead. So it breaks down fat to form fatty acids and glycerol. The pancreas actually has 2 main functions, which is digestion and assimilation. Assimilation, we'll talk about that in the next lesson, and this has to do with the hormone insulin that they release. But in terms of digestion, the pancreas, it releases the pancreatic juice to the duodenum, and that's its main role. The pancreas is not part of the alimentary canal. You can see that it's not part of the tube, but it's actually attached to it. So you call it an accessory organ. Now let's look into the function of the liver and the gallbladder. The liver is this big red part right here, and it is actually the largest internal organ. It is also described as the chemical factory. It's not part of the alimentary canal as well because it is not part of this big tube, but instead it is an accessory organ. Its main function is to produce bile. In terms of the digestive system, the liver actually has many more important functions. That's why it's called a chemical factory. So bile is actually made from red blood cells that are worn out. So what happens is that you all know this is a cross-section of a red blood cell, and that it contains a lot of these little red pigments called hemoglobin. So each hemoglobin molecule is made up of different groups. So you have the globin, which is this chocolatey color stuff, and this is the protein that gives it structure. And it's also made out of these 4 discs right here called the heme part. So this heme actually contains iron, and that's where you get the binding of oxygen molecule. So once the red blood cell has been worn out, so they're pretty old, you want to break it down in the liver. And liver does this, and it actually takes the heme when it breaks down the red blood cells. The heme group from the hemoglobin will actually form bile. So the gallbladder comes next after this, and what the gallbladder does is that after the liver makes the bile is temporarily stored in the gallbladder. So it's kind of like a bag, and it helps to store that bile. And you can see here that there's this long tube that actually is connected to the duodenum, and that is what we call the bile duct. So when we need when needed, bile will be released via the bile duct and then into the duodenum. So this bile is being made by the liver. But once we have once after we eat, we get the chyme, and the chyme actually enters the duodenum, and that's when bile is actually released so that it can emulsify the fat that's found in the food and the chyme, but also to neutralize the acid to provide an optimum environment for the other enzymes from the pancreatic juice and the intestinal juice to work on. So before we look into the next part, I actually wanted to show you guys how everything is kind of connected to each other. So this is a website called anatomylearning.com, and it can show you actually most of the organs and parts of the body. And here we have the diaphragm, and also right underneath the diaphragm is actually your liver right there. So liver is pretty huge, and you can see right down here is that there's the gallbladder. The liver also has like a vein called the hepatic portal vein, and also has an artery in there. But the most important part here is that we can see that it's connected to the gallbladder. So let's try to kind of like cut this apart. So maybe we can delete this segment here. So if you remove that, you can actually see that the gallbladder is not a part of the liver, but it is connected. So here you have like the duct right here that actually supplies the bile, and then the bile is stored here. So let's have a look at the gallbladder, and here is your gallbladder. So your gallbladder is connected to this here U-shaped organ called the duodenum. So right here, this is actually the pancreas, but they kind of made it a bit translucent so that you can see that this right here, this green bile duct is actually connected. So I'm going to remove that, and then you can see here that the bile duct actually connects to inside of the duodenum. So that's when you can release the bile into the duodenum. Let's try to cut and see what happens here. So let's slice it maybe this way. Okay, so you can see that it is a tube, but I actually wanted to see how that looks. So let's try to slice it like that instead. Okay, well, it doesn't show that it actually enters fully, but this is a very simplified diagram. So, okay, that's cool, that's cool. All right, where were we? Okay. So the function of the intestinal juice now. The intestinal juice comes from the duodenum. It is produced and secreted. Whenever you see the word secrete, it actually means released from the walls of the duodenum. This is where most of the complete digestion of nutrients takes place. So the intestinal juice is actually in charge of completing digestion. And when I talk about completing digestion, we mean that the nutrients are going to be the smallest of it and the most simplest form of that nutrient. So there are 5 enzymes found in intestinal juice. So this right here is a picture of what the duodenum would look like under the microscope, and you can see that this is the wall of the duodenum, and there are kind of like these folds right here. So there are also some glands around here which helps to make and release the enzymes. So that space right there, that's the lumen space. So let's move into enzyme back right here. So the first enzyme is intestinal lipase, and the substrate here is fat, and it produces the product is fatty acids and glycerol. So the equation that you can find here is fat being acted on lipase, and you get fatty acids plus glycerol. The second enzyme is erepsin, and this is a group of protease that can actually break down the polypeptide chains to produce amino acids. So the short polypeptide chains will then become its smallest form, which is amino acids. So let's have a look at how this occurs. Short polypeptide chains are actually made up of amino acid molecules that join together by bonds called the peptide bonds. So the group of enzymes called erepsin helps to speed up the breakdown of the peptide bonds to form individual amino acid molecules. And this is very small, and it's small enough to be absorbed into the body cells, and that's why we actually carry out digestion in the first place. So a short polypeptide chain right here is going to be acted on by erepsin. So what erepsin does is that erepsin will work on these bonds, and all three of those bonds will be broken, or even more, by erepsin to produce singular amino acids. The third enzyme is maltase, and maltase works on maltose to produce glucose and glucose. One molecule of maltose is made up of two molecules of glucose, and it's joined together by a bond called a glycosidic bond. Glycosidic bond is a type of bond that holds and combines two sugar molecules or even more together. The enzyme maltase can speed up the breakdown of maltose into two separate individual glucose molecules. So this is one molecule of maltose, and thanks to the maltase, it's going to break up that bond right there, and you get two individual glucose molecules separated. The fourth enzyme is called sucrase, and sucrase is also a group of enzymes, and a good example of sucrase is invertase. This enzyme acts on sucrose to produce glucose plus fructose. So one molecule of sucrose is made up of one molecule of glucose joined together with one molecule of fructose, and it is also connected by a glycosidic bond. But the enzyme sucrase can speed up the breakdown of sucrose into individual glucose and fructose molecules. This right here is glucose, and this shape right here is actually your fructose. And together, when they are bound together like that, you get sucrose. So the enzyme sucrase will act on that bond right there to separate it into glucose and fructose. Lastly, we have lactase. So this lactase actually acts on lactose, which is a type of sugar that you can find in milk. So some people actually don't have active lactase, so they become lactose intolerant. So they cannot break down lactose, and that's why they can't take milk very well. The product of lactase is glucose and galactose. So just like the other disaccharides, lactose is also made up of two different sugars. It's made up of glucose and galactose, and they both have a similar shape structurally, but there is a bit of differences, but we're not going to look into that. So I colored galactose as a purple here. So this is what we call one molecule of lactose. So what lactase does is that it's going to break up that bond right there to produce glucose and galactose being separated. So the walls of the duodenum actually have folds, and ones that you can see that there are folds here, and there are actually extra mini folds on them, and those are called the villus. We'll look at this into detail when we cover absorption, but right now we're looking at mainly digestion. So at the end of the duodenum, that's when the chyme is already being broken down. So once you actually look into that, so inside the chyme are actually these very, very small products right here. All the circle ones are amino acids, and those ones, the red ones, are glucose. We have galactose in purple here, and fructose right there in green. So at the end of the duodenum, all of the food molecules are in its simplest form, and it is soluble. And this is really good because it is now ready for absorption in the ileum. So the ileum is the end part, so the lower half of the small intestine. So if we just like have a look at the small intestine, so where is it here? So it's actually the ileum. So here, that part is the duodenum, and right after you actually get the small intestine part that is called the ileum, and this is where the main absorption actually takes place. And the ileum is very, very long, so that actually gives us a lot of time to fully absorb the nutrients that we need in our body. Now that's the end of our lesson for today. Thank you for watching, and I'll see you guys next time.